Dear students, today I am going to discuss about the topic dermatomycosis. Dermatomycosis is a medical term of fungal infections, mostly of the skin and the skin appendages. And this particular term has derived from the two words derma, that means skin, and mycos, that means fungi. This encompass nails and hair. This appendages mostly encompasses nail and hair. So fungal infection can happen anytime there are injured or pathologically changed skin areas. However, there are also factors which may promote fungal infections of healthy skin areas. Mostly warm, humid areas such as feet, spaces between toes, and armpits are often colonized by fungi. And due to this warm and humid environment, the conditions are optimal for the reproduction of the pathogen. Moreover, the skin is more sensitive, enabling the pathogen to enter the skin more easily. And it has been observed that approximately 70% of dermatomycosis, which are most commonly caused by the dermatophytes, the infection that is passed on from person to person or via contaminated object, or maybe from contaminated animal to human. Dermatomycosis are mainly caused by the filamentous fungi. These are the fungi that feed on keratin occurring on skin, hair, and nails. And these fungi, there are three different types of fungi: anthropophilic, zoophilic, and geophilic. So this anthropophilic dermatophyte, that is the Trichophyton rubrum, this particular species, fungal species, is still the most frequent causative agent of fungal disease worldwide. And here the virulence factors are keratinolytic enzymes and cysteine dehydrogenase. This keratinolytic enzyme is actually the two enzymes present there. One is hydrolase, another one is keratinase. And recently, the third enzyme, that is cysteine deoxygenase, that has been observed as a new virulence factor, influencing host factors by playing a similarly important role for the development of dermatophytosis of the skin and nails. So filamentous fungi that can be transmitted from human to another human by close contact with an infected human being or infected animals. Some pathogens are mainly found in the soil, so that they can be transmitted during gardening. Besides filamentous fungi, in rare cases also yeast and molds may cause dermatomycosis. Generally, infections with the mentioned pathogens can occur on all body parts. The severity of infection depends on the causative agent or agents. And mixed infection with different pathogens occur frequently. So chronic venous insufficiency, diabetes mellitus, disorders of cellular immunity, and genetic predisposition should be considered as risk factors for onychomycosis, snail infection. A new alarming trend is the increasing number of cases of onychomycosis, mostly due to Trichophyton rubrum. So there are four classes of four basic groups of fungal diseases. These are keratomycosis, dermatomycosis, candidiasis, and systemic mycosis. Today I'm going to focus about dermatomycosis caused by epidermophyton, trichophyton, mycosporum, rubrophytes, as well as pappus. And this is the most widespread group that is causing fungal disease. So, dermatomycosis, that is divided according to the responsible pathogen into, into dermatophytes, yeast, mold infection, fungi on the skin or dermatophytes are the cause of dermatophytosis and also referred to as tinea or ringworm. The most common dermatophyte and probably worldwide is Trichophyton rubrum. They are causing infections throughout the body, in skin, in head, uh, in hair, in nail. And this nail infection is known as onychomycosis, a very common disorder in industrialized nations. The prevalence of fungal nail infection among certain risk groups, those who are diabetic, is higher than the general population, and it is also higher in patients with disorders of uh, keratinization affecting the skin and nails in case of psoriatic patients. The clinical diagnosis is often difficult given that nail disorder due to other causes must be ruled out. A new epidemiological development is the increase in onychomycosis, usually due to Trichophyton rubrum. So, the study of dermatomycosis is of great epidemiological importance, while the organization of their control is a problem of social significance. Now, I'm coming to the dermatophytes. So, the dermatophytes, on the basis of their natural habitat and host prevalence, can be classified into three different groups: anthropophilic, zoophilic, and geophilic. Anthropophilic dermatophytes species are typically adapted to live on human host. As for example, Trichophyton rubrum, Mycosporum, Odoni, etc. etc. Epidermophyton flucusum, like that. Zoophilic species, they live on animals and often cause infection in their animal host. Examples are Trichophyton, Violacea, Mycosporum, Canis. And the third one, that is the geophilic species. The geophilic species are saprophytic fungi that are found in soil or in humus. They occasionally cause infection in human and animal. Examples are Mycosporum, Gypsium, Trichophytum, Eloi, like that. The most important dermatophytes that cause infection in human are classified into these three genera: Trichophyton, Mycosporum, and Epidermophyton. Trichophyton, they are thin-walled, smooth, they have 4 to 6 septa and causes infection in hair, skin, and nail. In microsporum, they are thick-walled projections and 5 or more septum, and they cause infection of hair and skin. Then epidermophyton, they are also thick-walled, pear-shaped or oval-shaped, have 4 or fewer septa and causes infection in skin and nails, but not in hair. So these are the etiologies of dermatomycosis. Now I'm coming to the mode of transmission. So mostly the dermatophytes transmit by direct contact with infected host, and that infected host can be human or animal. By direct or indirect contact with infected exfoliated skin or hair in clothing, comb, hairbrushes, theater seat, caps, furniture, bed linings, shoes, socks, towel, hotel rugs, bathhouse, locker room floors, etc., etc. They can cause infection, is fungal infection to an individual. Next one is, I'm coming to the 3 species. That is, first I will start with Trichophyton. So here you will see how the colonies will look like. They show color. The conidia is sparsed, means they are discrete, very loosely bound, thin-walled, smooth and septate, pencil-like structure. In case of microsporum, you can see the lactophenol cotton blue stained culture, and mostly from the scalp lesion we can get this type of fungal species. And we can see bright green appearances of the infected hair under the UV light only because of microsporum infection. They form microconidia, which are numerous, thick-walled, rough, and you will see their spindle side. Now, pathogenesis of Trichophyton. So far, 21 species have been identified that can cause trichophytosis, means infection of hair, skin, and nails. Transmission took place on the sick people and things of general use. Superficial trichophytosis of the scalp is caused by a species which is known as Tinea capitis. And the most common agent is Trichophyton rubra. These are of 2 different types. One is known as endothrix and second one is ectothrix. Endothrix means those who grow entirely within the hair shaft and break off the hair. Trichophyton violaceum is a species, and this picture will tell you what are the endothrix. So here you will see they are growing internally at the shaft, hair shaft. And next one is the ectothrix. They grow around but outside the hair. And here you will see at the surface of the hair they are growing. These are known as an ectothrix. Trichophytum rubrum is an example. Both the ecto and endothrix fungi destroy keratin by keratinolytic enzymes, and giving the hair is a broken hair appearance. Next one is the microsporum. So to date, 17 species have been identified which cause infection in hair and skin, not in nails. Penetrate the hair and extend down shaft as fine filament. They belongs to anthropophilic as well as zooanthropophilic fungi. Their epidemiology is pretty much similar with trichophytosis, and often they infect the scalp. And here you will see this roundworm like appearance caused by this particular species. Next, the third one is Epidermophyton. So this is the cultured colony of Epidermophyton flucusum. It is whitish yellow or olive in color, and under the microscope via lactophenol cotton blue staining, you can see this smooth-walled, thick-walled, club-shaped macroconidia with 4 to fewer septum that is present here of Epidermophyton flocosum species. And this particular species, they are actually superficial or cutaneous mycosis, the cause include in this flocosum. So they causes four different type of infections. The ringworm caused by Tinea corporis. This is how the ringworm look like. You can see this central part and this raised edges of the ringworm or the lesions in the skin. Then jock itchy skin is caused by Tinea cruris. Athlete's feet is caused by Tinea pedis, and fungal infection in the nail is caused by Tinea anguinum. So this is all about uh, epidermophyton infection. The next one is rubromycosis caused by Tinea rubrum or Trichophyton rubrum. It affects not only the skin but also the hair and is a highly contagious and widespread fungal species. The transmission is by the same way as in the case of epidermophytosis, and we have to pay attention of sharing towel, gloves, and be aware of handshake. So we have to restrict handshake. Last one is Favus. The causative pathogen or causative agent is Trichophyton swainleni, which is an endothic species. And these are less contagious but cause chronic infection. Infection from sick people and through things. Children are most often get infection by this particular fungal pathogen. Usually the hairy part of the head and rarer the nail plates or the and still rarer the skin are getting infected by this particular species. Mostly the weak children, they frequently get infected by Trichophyton swainleni, and here the infection can also spread to the internal organs like lungs, digestive tract, and even the brain. So diagnosis is based on typical clinical features and is also confirmed by the laboratory findings. So how the diagnosis will occur in case of this fungal pathogen? So mostly diagnosis, laboratory diagnosis is based on demonstration of fungal elements in clinical specimen by microscopy and contamination by culture. Confirmation by culture. The specimen includes skin scrapings and nail clippings, or maybe hair taken from the areas suspected to be infected by dermatomycosis. So these entire specimens are treated with alkali solution, 10 to 20% K solution, potassium hydroxide solution is here to clear epithelial cells and other debris. Then direct microscopy is usually done for the diagnosis. While culture is always carried out to identify the causative fungal agent, and along with that, when we are going for culturing, the specimen is inoculated on subodextrose agar plate containing the two antibiotic chloramphenicol and chlorhexamine. So these two antibiotics will prevent the growth of bacteria and saprophytic fungi. So after culturing into the SDA plate, the incubation temperature would be 25 to 30 degrees centigrade, and you have to culture for at least 2 to 3 weeks. So there you will get the fungal colonies, and the colonies. So here for culturing, uh, we have to take this the antibiotics we have to use, and then when there is regular interval, we have to check, we have to examine, we have to check the dermatophyte appearances, and by monitoring the colony morphology, pigment production, and also the presence of macro and microconidia, we can classify, we can detect which pathogen it is. Like only macroconidia are present in in epidermophyton infection. Few macroconidia and more microconidia are present in trichophyton infection. Macroconidia are predominantly present in microsporal infection. So the differentiation of the three genera is based mainly on the nature of macroconidia. So this is how the Trichophyton rubrum species look like. One is the milky white thallus structure,